Today we're going to be talking about replication, and I have chosen to go with a little bit of a flashback style here. My sister is an artist, so I've stolen some of her drafting paper, and I'm here in Fort Robinson, Nebraska, kind of doing this rustic style. So what I want to talk to you about today is the process of DNA replication, and I'm just going ahead and drawing a very simplistic depiction of a bacterial chromosome or plasmid, recalling that, of course, they're single circular molecules. Now, we can make both strands of DNA, and remember that they would be, of course, supercoiled, and they would also be in their double helical variation. But if you could use your imagination with me and pretend that this was a ironed flat out on the page version of a DNA chromosome or plasmid. Now, just for sake of giving a specific example, let's say that this is the E. coli chromosome. So we're just going to call this the E. coli chromosome, single circular piece of DNA. And we know that on this E. coli chromosome, there is an origin of replication. So right here is that area that we typically call the OREC, or the origin of replication. Now, this is where all of it is going to begin. So this is the region at which we're going to need to see the binding of two replisomes. Replisomes are complexes of upwards of 30 enzymes that are required to replicate or make a copy of the DNA. And so I'm just going to draw one replisome here, and then we've got another replisome here. And these two replisomes, they kind of back up next to one another. And they put their backs to each other right there at the origin of replication. And this replisome is going to go this way around the chromosome, and this replisome is going to go this way. And as you can see, that really indicates that they're going to meet at some point on the other side, directly opposite. Let's see how well I did there. Maybe more like here, right? Directly opposite of where the origin of replication is. And at that site, this is called the termination sequence. And the termination sequence is going to be recognized by an enzyme called the terminator utilization substance, or TUS. Now, every time I think of TUS, all I can think of is Arnold Schwarzenegger saying, "I'll be back." So the terminator utilization substance. So what happens is when these two replisomes hit this opposite side, that's where replication is terminated. Now, in the meantime, of course, both of these replisomes synthesize two new strands of DNA. So this process is a bidirectional, right? Two directions, one going in one way, one going the other way. It's a bidirectional process. It is also semi-conservative, meaning that at the end, each of the new molecules of DNA, that each of the new E. coli chromosomes that are made will contain one strand of new and one strand of old. So it is a very semi-conservative process. Now, in order to see that happening, we're going to have to move our picture just a little bit. So in order for these replisomes to proceed along their way, of course, we know that the DNA has to become the supercoiling has to be relieved. That's done by an enzyme called topoisomerase. And additionally, we know that it has to be unzipped. So the epic pickup line of "I wish I were DNA helicases, so I could unzip your genes." It's DNA helicase is the enzyme that'll do that. So these replisomes don't have everything that they need, but have to be assisted by some other enzymes. Now, we're just going to go ahead and draw what happens, though, at this origin of replication. So as we return to the origin, so we're returning to the origin of replication, only this time we're showing a little bit of a time lapse that has taken place after the two replisomes have bound. So this is after the a region of the DNA has been unzipped by DNA helicase. And we now have one replosome that has headed a little ways around here, and another one here. Oh dear, drawing skills. My sister's gonna have a fit when she sees this in her drafting paper. So we have these two replisomes have parted ways and they've begun their synthesis. So the replosome that is headed in this direction and the replosome that is headed in this direction, both of them are making two new strands of DNA. So this one is creating these two strands. And of course, this new strand here is complementary to this existing or old strand here. This new strand is complementary to this one. And likewise, this replosome is also synthesizing two new strands of DNA. So it also is making two strands. Now, of course, there has to be a DNA ligase enzyme involved to seal the little nick that was there at the start. And so we get two very important terms coming up. This right here, where the one replosome is headed this way, it's creating a fork. This is called a replication fork. So this is one replication fork right here. And this over on this side, in this area, see that forking there, fork in the road, right? This is another replication fork. Now, as a whole, this here of the new DNA is termed a replication bubble. And you can see why it's kind of looking like a little bubble there. So that is termed the replication bubble. Now you can see how as both replication forks go in their opposite directions, that replication bubble is going to get bigger and bigger and bigger. So let's do another time lapse and show the replication bubble getting bigger. So now we're seeing this replication bubble getting much larger. So I'm just going to draw much more time has lapsed. Here's our replication fork on this side, and likewise a second replosome. So replisomes heading again in both ways. getting close to the terminator utilization substance that's recognized the terminator site or the termination site on this side. And so now the bubble has become fairly expansive. And if you can use your imagination now with me, that we are looking at, okay, so remember that this replosome made this strand, and it made this strand, and this one made this strand, and this strand. And now we've opened up an entire region here where if you imagine you're almost looking at that parting and opening where the replication bubble is consuming much of what has been the original chromosome. And now eventually that's going to get so large that it will look like there's only a tiny piece knit together. And one thing that's very important to recognize on each one of these, and if we're almost looking down on that, is that now the OREC on both of them has now been again exposed. And so another round of replication can actually begin. So you can actually see two more replisomes binding at this origin of replication and also binding here at this one. And so this enables literally the E. coli chromosome to be getting replicated before the first round of replication has ended. The second round of replication has literally begun. So now let's take a moment to look at what it would look like basically at the end of our process of replicating the chromosome, at least for one time. Now, at this point, the replication bubble has become so big that essentially it seems like we basically have two separate new chromosomes. But let's just remind ourselves that we've still got our replisomes. There's one there. It's headed this way, and one here. It's headed that way. They've basically are on top of the terminator site or the termination site right here where they'll meet with pus. So these are almost ending replication. And of course, in both cases, we had the strands that were synthesized. So this replosome had synthesized from the start, from where our initial OREC was. It had synthesized this one, and it had synthesized this one. See, now they're like pulled apart. Essentially, we saw the bubble beginning to pull apart, and now it's like basically pulled entirely apart. And then this one had synthesized this strand, and it had made this one. And so, of course, remember those had been ligated together. So we now see what is basically an all-encompassing bubble to the point where we almost have two independent copies of the chromosome. And as soon as those two replisomes hit the termination site, then those will pull apart, and we will have now two separate copies of the initial E. coli chromosome. So remember that I had mentioned that another round of replication could begin even before this one ended. And I want you to keep that in mind. I actually want you to do a calculation. So this is something I want you to bring with you and have it be a product of your pre-partying. So if we know that the E. coli chromosome is 4.6 times 10 to the 6th base pairs, Or we usually say that a little bit differently. We usually say that it's 4,600 kilobases by making it easy for you by saying it's 4.6 times 10 to the 6th base pairs. And we know that the primary polymerization enzyme in the replosome called DNA polymerase. So DNA polymerase is an epic enzyme within a multi-enzyme complex. DNA polymerase can work at epically fast rates, 1,000 base pairs per second. So I want you to use that information, and I want you to calculate how long it would take for E. coli to replicate its chromosome. And using those numbers, I want you to scratch your head and say, why doesn't that make any sense? And bring that with you as a product of your pre-party. Bring it with you to class. Okay, we need to talk a little bit more about the epic DNA polymerase. Now, in order to talk more about DNA polymerase, we need to zoom in. So DNA polymerase is one of the enzymes in the replosome, one of a multi-enzyme complex. And I want to literally zoom in on one region of the replication fork, and I just want to look at this one region. So we're going to do this kind of massive zoom in this area, where we're going to note this as a 5' end on the DNA that we're kind of cutting off right here. And that'll be the bottom of our zoom in. But then we've got this whole area, and I'm just going to sketch that this is like all this open area. Now, of course, this strand has to be anti-parallel, so this is the closed region here. It still hasn't been, DNA helicase hasn't gotten hold of it. It hasn't unzipped the genes here in this area, but this whole area is open. Okay, it's unzipped. Now we know we've got two strands that are getting made, and let me kind of draw in our replosome. So it's essentially this huge, now looking very large. And I'm just drawing it lightly because it's not really the focus of what we want to look at here. And so we know that DNA polymerase is an enzyme in that replosome, synthesizing the strands of DNA. And I actually just want to call your attention to this strand of DNA. So we know this replosome is making two. We recognize this one is necessarily anti-parallel to this one. So what I want to look at now is understanding what DNA polymerase needs in order to build this new strand of DNA. So if we're just focusing on that new strand of DNA, this is going to necessitate us making another zoom. So I actually want to just look, if we were to zoom in a very small region here, zooming out from here, and I just want to look at, here's this top strand, and I want to say that we actually know the sequence. So there's a 3' side, and if this is, the nitrogenous base is T, a G, C, A, T, okay, in that region there. And then, of course, as DNA polymerase, right, the enzyme in the replosome, as it makes this new strand of DNA, it's pairing with that one. So anti-parallel, and then T is going to bind to A, G to C, C to G, A to T, T to A, right? And it's building in only one direction. That is, DNA polymerase is only capable of building in the 5' to 3' direction, adding new nucleotides to the open 3' hydroxyl. We could actually literally show its structure, recognizing that, and actually let's, yeah. This is an A, and then at the carbon 5, there's an attachment to a phosphate. And then that keeps on going. Well, this right here, remember this is DNA, so the 2' carbon is got a hydrogen, right? It's deoxy at the 2' carbon. But right there is the hydroxyl. The building takes place, so right here, if this is the most recent nucleotide right here, then the next nucleotide, and in fact nucleoside triphosphates are the building blocks, so the next nucleoside triphosphate to be added will be added right onto that 3' hydroxyl right there. So DNA is built in the 5' to 3' direction, adding onto the 3' hydroxyl as the building takes place. Now, it's important to note that in order for DNA polymerase to build, it requires an open 3' hydroxyl. So that is to say, if we zoom back here to the very start, the very, very start of this strand of DNA that this replosome is making, it had to have had a 3' hydroxyl to start from. That is to say that DNA polymerase, why it is a very, very fast enzyme, is very, very needy. It requires a lot of things. First off, it requires a primer, so there had to have been a little tiny piece of nucleic acid right here to kick things off for DNA polymerase. And in fact, because DNA polymerase can't start without that open 3' hydroxyl, it has to rely upon RNA polymerase to make a tiny little RNA primer at the very beginning of the synthesis of the DNA. So RNA polymerase can start cold without a 3' hydroxyl, so it puts together some RNA primers, ones with hydroxyls there, and then later it goes back and swaps those out for DNA so that it provides a primer. So DNA polymerase is a super needy enzyme. So I want to write down the things that DNA polymerase requires. So DNA polymerase requires several things. It's a very fast but very needy enzyme. We know people like that, right? Number one, it has to have a primer. And by saying it needs to have a primer, what we're saying is it has to have an open 3' hydroxyl. So an RNA polymerase has to provide that for the DNA polymerase. So that open 3' hydroxyl is often provided by an RNA polymerase called primase that lives inside of the replosome. Remember, more than one thing living inside of that replosome. So it also, number two, it's going to require the building blocks. So as it continues to build, like when it went from having just paired this G with a C, then it needed the next building block, which was another G. But we know it's more than just the nitrogenous base, right? It's the full deoxynucleoside triphosphate that was needed next. So the building blocks of DNA are called just broadly DNTPs, deoxynucleoside triphosphates with 3 phosphoryl groups attached here, and then the hydrolysis of those leaves just one attached, and then of course that one attaches onto the next 3' hydroxyl and so on forth. So DNTPs are of a couple types. There of course are DCTPs, DGTPs, DTTPs, and DATPs. So it requires building blocks. And then lastly, of course, it was this template strand, the original strand, that told the DNA polymerase what to pair with. So it needs a template. Awesome. Now, you'll notice that when I made this drawing here, I left out the other strand that this particular replosome makes. And that was intentional, because if you go back and look at that strand, three prime end here, 3' end here, meaning that this strand appears to have been made in 3' to 5' direction. But that is impossible. That cannot take place, because we know that the new nucleotides, as they're added, are added to the open 3' hydroxyl, not to the phosphate, the 5' phosphate. And so synthesis in the 3' to 5' direction simply cannot, does not, absolutely does not occur. So in order to actually look at what does occur on this particular strand, this strand here that is synthesized easily 5' to 3' is called the leading strand. Now this strand that appears to be synthesized 3' to 5' is called the lagging strand. But the lagging strand can't be synthesized that way. So now what I want to do is let's redraw this in the right way, showing how the lagging strand actually gets synthesized. So once again, we'll take our attention to this particular replication fork. No problem for the leading strand, 5' to 3'. But this lagging strand cannot get synthesized 3' to 5'. So what literally has to happen is that this strand literally has to turn around this way and go into the replosome backwards in order to be synthesized in the 5' to 3' direction. But recall that, of course, that couldn't happen the whole time, right? So instead, this actually happens in short spurts. It's like it goes in backwards, there's a little spurt, and then it lets go, and then there's a little spurt. So it's actually synthesizing in the opposite direction. So it's like this, right, actually synthesizing backwards on this this other on the lagging strand. So in order to support that, of course, each spurt has to have a primer, right? There has to be a primer. So there's a primase enzyme in the replosome, and as the leading strand is being made and going that way, the primase, the RNA polymerase in the replosome, is quickly synthesizing short spurts of RNA on the lagging strand. And of course, these have open 3' hydroxyls on them right here. It's an open 3' hydroxyl. So now what can happen is when when this strand gets turned back around and fed into the replosome that direction, now this short spurt from the open 3' hydroxyl can get made like that, like that, right? So these right here are called Okazaki fragments. Now, we also know that there's got to be a removal of these little RNA primers, because of course these are RNA. So there's another DNA polymerase called DNA polymerase 1 that comes through and removes those, and it replaces them with DNA. So eventually these become DNA, which means that we almost have what looks like a 3' to 5' strand, except for that DNA ligase has to seal the nicks, making the 3', 5' phosphodiester bonds that need to form right there. So in the end of the day, this replosome has essentially synthesized both a 5' to 3' leading strand and a 3' to 5' lagging strand.